So hello students and welcome back again to another video tutorial by Genome Biotech. In this video tutorial, we would be talking about the DNA polymerases. DNA polymerases, as you know, are the main replicating enzymes that synthesizes a new DNA strand on a template strand by the process of DNA polymerization. So this is the main replicating enzyme that is responsible for the biosynthesis of the DNA molecule. So DNA polymerases are the enzyme that synthesizes a new DNA strand over a template strand that by the process of polymerization is referred to as DNA polymerases. Now, DNA polymerases have been categorized into several different types. For example, the DNA polymerases in the case of E. coli that we are going to talk about has been categorized into the following types. The DNA polymerases in E. coli can be DNA polymerase 1, DNA polymerase 2nd, DNA polymerase 3rd, and so on. DNA polymerase 1 is also referred to as the Kornberg DNA polymerase. The most important DNA polymerase among all is the DNA polymerase number 1, which is also referred to as the Kornberg DNA polymerase. This is the Kornberg DNA polymerase, okay, who subsequently won the Nobel Prize for his work. It was Kornberg who subsequently won the Nobel Prize for his work. Is primarily utilized in in filling the small DNA segments during replication and repair processes. So DNA polymerase 1, which is also referred to as the Kornberg polymerase, as it was being discovered by Arthur Kornberg, is a repair polymerase that fills in the gaps that are being produced due to damage over the DNA molecule. Now, when the DNA gets damaged, there are gaps which are being produced in the DNA. Those gaps are being filled up with the help of DNA polymerase 1 enzyme, which is also called as Kornberg polymerase. Okay. DNA polymerase 2nd is another enzyme that serve as an alternate repair polymerase. If the DNA polymerase 1 is not going to function, then there is the next enzyme that is going to be functional, and that is referred to as DNA polymerase 2nd is the second enzyme that is going to be functional. It can also replicate DNA if the template is damaged, and the last enzyme is referred to as DNA polymerase 4 or DNA polymerase 5. Now, DNA polymerase 4 and DNA polymerase 5 are quite unique enzymes. They are known as Y family DNA polymerases. They are exceptional. They are exceptional because they do not require any template for their own biosynthesis of nucleotides. These DNA polymerase 4 and DNA polymerase 5 works in a template independent manner. These DNA polymerases always works in a template independent manner. These DNA polymerases, DNA polymerases, polymerases works in a template independent manner. Works in a template independent manner. It is the one that is going to be working in the template independent manner. There's no template whatsoever is required for the action of the Y family DNA polymerase. These DNA polymerase are functional in a repair system, and that repair system is referred to as SOS repair system. SOS repair system. Save Our Soul repair system is a type of repair system that utilizes the DNA polymerase 4 as well as utilizes the DNA polymerase 5, because both of these are the DNA polymerases that would be used in such type of a polymerization system, and they are referred to as the Y family DNA polymerases. These are called as the Y family DNA polymerases. Any doubt, any problem? Okay, now let us understand firstly about the DNA polymerase 1, that what is this DNA polymerase 1, How does the DNA polymerase 1 enzyme act? In what manner this enzyme is going to be functional? So let us look at this DNA polymerase 1 enzyme. Okay. To have a view of this DNA polymerase 1 enzyme, let me just go and make you visible about the DNA polymerization reactions and about the molecular biology notes. Okay, so here we are. We are now going to talk about DNA polymerases. So where is DNA polymerase? Here it is. DNA
In order for
polymerase.
genetic information to be passed.
Now, let me just show you the animation so that you can visualize the same. Now, in order for the genetic information to be effectively passed from one generation to the other, it has to be copied. And that copying is being performed with the help of an enzyme that is called as the DNA polymerase. The DNA polymerase that we are going to talk about here is referred to as the DNA polymerase 1, which is also referred to as the Kornberg DNA polymerase. So this is the DNA polymerase 1, DNA polymerase 1, also referred to as Kornberg DNA polymerase. Polymerase. Okay. This is the Kornberg DNA polymerase for which we are going to talk about. Now, how does this DNA polymerase is going to function? That we are going to look forward. So please have a view.
Passed on from a parent cell to a daughter cell, the parental DNA must be copied into two identical daughter DNA molecules. When you have completed this exercise, you should understand the chemistry of DNA synthesis, understand the mechanism of catalysis by DNA polymerase, and understand the proofreading function of DNA polymerase.
Okay. So we must understand the chemistry of the DNA synthesis, that how the chemistry of the DNA synthesis actually takes place. What is the chemistry of DNA synthesis? In which manner the DNA is being biosynthesized in the cell? Understand the mechanism of catalysis by DNA polymerase. How the catalysis is being performed by the DNA polymerase. How DNA polymerase catalyzes the reactions. Understand the proofreading function of the DNA polymerase. That how DNA polymerase proofreads the DNA molecule and in what manner the proofreading is going to be performed, how and in what manner the proofreading of the DNA is going to take place. So let us have a view.
DNA synthesis requires deoxynucleoside triphosphates and a primer template junction.
Okay, so whenever you want to initiate the DNA biosynthesis, you require a primer molecule. Okay, you require a template and a primer molecule. So this is the template which is being provided over which the primer is going to bind. The primer, once it is being bind to the template, will have a hydroxy group present over itself, and that would be the 3' hydroxy group. This 3' hydroxy group would be extended with the help of DNA polymerase enzyme, and the DNA polymerase enzyme is going to extend this 3' hydroxy group indefinitely.
Each of the four DNTPs, DGTP, DCTP, DATP, and DTTP, has three phosphoryl groups which are attached via the 5' hydroxyl of the 2' deoxyribose. The phosphoryl group closest to the ribose is the alpha phosphate, the middle group is the beta phosphate, and the outermost group is the gamma phosphate.
Okay. So there are three different phosphates which are present. One is called as the alpha phosphate, the second one is the beta phosphate, and the third one is referred to as the gamma phosphate. These are the three phosphate molecules which are present, and the innermost alpha, beta, and gamma. So those are the three phosphate molecules which are present, and it is this molecule is present in the form of deoxynucleotide triphosphates. You can find this is nucleotide triphosphate, three phosphate molecules, but this nucleotide triphosphate molecule is going to react, but the incorporation of only nucleotide monophosphate is going to take place. Incorporated nucleotide will be only monophosphate, never di or triphosphate molecules. Please have a look here.
The primer template junction has two key components. The template provides the single-stranded DNA to be copied. The primer provides a free 3' hydroxyl at which a DNTP can be added.
Okay, so see this. So DNTPs can be added to the free 3' hydroxy group which is being provided by the primer molecule. So to this primer 3' hydroxy group, the nucleotide monophosphate, nucleotide triphosphate will be making attack. The triphosphate will once make an attack, then this hydroxy group will attack the triphosphate molecule in such a manner that nucleotide triphosphates will come and monophosphate will get incorporated. How?
The 3' hydroxyl of the primer strand attacks the alpha phosphoryl group of the incoming DNTP in a
Now see this. In a nucleophilic acyl substitution reaction, the hydroxyl group, which is present at the 3' end of the sugar, that is the 3' hydroxy group, will lose its hydrogen, will have an oxyanion, that is oxygen negatively charged molecule, will make an attack on the phosphodiester backbone. See, this is what is going to happen. O will have a charge that is negative, sorry, oxygen will have a charge that is negative. After forming an oxyanion, this will make an attack over the innermost phosphate molecule. The beta and gamma phosphate molecules will be broken down. Once the beta and gamma phosphate molecules will be broken down, then this beta and gamma phosphate molecules will be released. And once the beta and gamma phosphate molecules will be released, then these gamma and beta and gamma phosphate molecules released will liberate very large amount of energy here. When it will liberate very large amount of energy, then what will happen? As the large amount of energy will be released, so they will catalyze the reaction to take place by itself, clearing all the drawings and moving forward.
nucleophilic substitution reaction. The leaving group for the reaction is pyrophosphate, which is formed from the beta and gamma phosphoryl groups of the DNTP.
Now, the leaving group being pyrophosphate, which has been produced, this pyrophosphate is going to undergo hydrolysis. When this pyrophosphate is going to undergo hydrolysis, it would be lies down and energy will be released.
The free energy change for this reaction is rather small. However, additional free energy is provided by the rapid hydrolysis of the pyrophosphate into two phosphate groups. This reaction is catalyzed by the enzyme pyrophosphatase.
Okay, the reaction is being catalyzed by an enzyme, and the enzyme is referred to as pyrophosphatase, which is going to work in this particular manner, and then the energy will be liberated. Next point.
The process can then be repeated, with the 3' hydroxyl of the newly added DNTP serving as the nucleophile. The chemistry of DNA synthesis requires that DNA be made in a polar fashion. In biological polymerization, DNA is always made by extending the 3' end of the primer strand.
Okay, in the biological polymerization reactions, the DNA can be extended by extending the 3' end of the DNA molecule. So the DNA molecule 3' end can be extended to a greater length, making the complementary copy of the DNA molecule.
The template strand directs which of the 4 possible DNTPs is added. During replication, the DNTP that base pairs with the template strand is highly favored for addition to the primer strand.
Okay, now there are template strand direct which DNTPs is going to be added through base pairing. So which nucleotide triphosphate is going to be added is being determined by the template strand. This is the template strand. It will govern that which nucleotides will be joined and where these nucleotides are going to be joined and how. Next.
The main enzyme of DNA synthesis is DNA polymerase. The DNA substrate sits in a large cleft of the DNA polymerase that resembles a partially closed right hand. Based on the analogy to a hand, 3 domains of the polymerase called the palm, fingers, and thumb have been described.
Okay, there are several different domains which have been described. One is referred to as the palm domain, the second one is referred to as the thumb domain, and the third one is referred to as the finger domain. So these are the three different types of domains that has been described in the structure of the DNA polymerase 1, which is also referred to as the Kornberg polymerase. Next.
The palm domain houses the active site for DNA synthesis. The active site of DNA polymerase is able to distinguish between RNTPs and DNTPs, even though RNTPs are present at approximately 10-fold higher concentration in the cell. The nucleotide binding pocket is too small to accommodate the presence of a 2' hydroxyl on the incoming nucleotide, allowing the polymerase to sterically exclude RNTPs.
Now, this is very important. See, nucleotide binding pockets is too small for the 2' hydroxy group on the incoming ribonucleotide to be incorporated. The incoming ribonucleotides cannot be incorporated in the pocket because it cannot accommodate the 2' hydroxy group. And you know that 2' hydroxy group is only present in the case of ribonucleotides. So ribonucleotides are going to be expelled out. So ribonucleotides will be expelled out from this particular location, and only the deoxyribonucleotides will be incorporated. This is the deoxyribonucleotide. How?
Correct base pairing is also required for catalysis. If an incorrect base pair forms, the alpha phosphoryl group of the DNTP cannot properly align with the 3' hydroxyl of the primer strand. Once the proper DNTP
There was no alignment, no proper alignment that was there between the alpha phosphate and the hydroxy group, and the 3' hydroxy group. So the alignment was not proper. The bonding was not there because it was not the complementary nucleotide strand. So such type of nucleotide strand will be lost.
is bound in the pocket, the reaction can continue.
Okay, so if the correct nucleotide is going to be bounded in the pocket, the reaction will continue, and here the reaction will take place and the phosphate molecule will be joined.
The palm domain also binds two divalent metal ions. These metal ions are crucial for DNA polymerization activity.
Now, we are all present still in the palm domain of the DNA polymerase first. The palm domain also contains two metal ions. One is referred to as the metal ion A, another one is referred to as the metal ion B. Both metal ion are going to reduce the forces of repulsion in between the phosphate molecules. When the forces of repulsion in between the phosphate molecules are going to be reduced, then the phosphate molecules can effectively remain attached to the palm domain, and the interaction of the pre-existing template nucleotide with the incoming nucleotide further elongates the binding of the incoming nucleotide with the template. This provides the opportunity for the 3' hydroxy group to attack the innermost phosphate. This provides an opportunity for the 3' hydroxy group to attach the alpha phosphate and forms a bond with it.
Once a correct base pair is formed between the incoming DNTP and the template, the finger domain moves to enclose the DNTP. This conformational change brings the divalent metal ions into the correct positions to function.
Okay. Now the finger domain moves. Once the finger domain is going to move, this is the palm domain, and this is a finger domain. The finger domain once moves, it brings the phosphate molecules in close proximity with the 3' hydroxy group, as well as initiate the conformational changes, bringing the DNTP and the primer into correct orientation with metalines, so that the primer and the DNTPs are being brought together at correct orientation. The primer last nucleotide can bind with the incoming nucleotide and forms a phosphodiester bond.
Metal ion A helps to deprotonate the 3' hydroxyl of the primer, producing an oxyanion to attack the alpha phosphate of the incoming DNTP.
See how the reaction is going to take place. Firstly, an oxyanion has to be produced. For the production of oxyanion, the hydrogen atom is scavenged, or it has been absorbed from this hydroxy group by the metal ion. So this hydrogen atom is being snatched, and once the hydrogen atom has been snatched, then there is an oxygen negative atom, and this oxygen negative is going to attack the innermost phosphate.
Metal ion B coordinates the negative charges of the beta and gamma phosphates of the DNTP and stabilizes the pyrophosphate leaving group. Lysine and arginine residues on the finger domain also help to stabilize the pyrophosphate. And, through stacking interactions, a tyrosine residue attached to the fingers helps to hold the DNTP in place for catalysis.
Now, before we move forward, please look here. The lysine, arginine, both of these are basic amino acid residue. As these are basic amino acid residue, so they will be binding to the negatively charged phosphate molecule. Lysine and arginine being basic will bind to the negatively charged phosphate molecule. As they will bind to the negatively charged phosphate molecule, so they will stabilize the negatively charged phosphate molecules. Then the incoming nucleotide has been also stabilized with the interaction with the tyrosine residue from the finger domain. The tyrosine residue helps to hold the deoxy nucleotide triphosphate in a correct orientation. Last, metaline A helps to deprotonate the 3' hydroxy group of the primer. So metaline, which has deprotonated the 3' hydroxy group, this 3' hydroxy group is going to attack to the phosphate molecule, and then there is the phosphodiester backbone which is being produced. Metaline B coordinate negatively charges of pyrophosphate. These are the negative charges of the pyrophosphate which are being coordinated by the metalines. Next, there is the bond which is being produced.
The finger domain also plays a second role. The finger domain associates with the template region, leading to a nearly 90-degree turn of the phosphodiester backbone of the template immediately after the active site.
Now see this finger domain. Now we have, or we already know that the active site where the entire reaction is going to take place is the palm domain. Okay, this is the domain. The second is the finger domain. Finger domain is helping to bring the phosphate molecules to their correct orientation so that the reaction can take place in between the incoming nucleotide triphosphate and the pre-existing template. Now, there is a turn that has been introduced in the helix of the DNA. You can see this black color line indicates the helix of the DNA. This is the helix of the DNA. This helix of the DNA has brought a turn, and that turn is of 90 degrees. See, this is the turn. Once the turn has been made, then the angle which this DNA makes is 90 degree. 90-degree turn of template helps avoid confusion in the active site, that where is the active site, so that only a single nucleotide is being exposed at the active site of the DNA molecule. Okay, now this DNA single nucleotide will be copied in the form of its adjacent nucleotide. Okay, so another nucleotide will be incorporated here only, and slowly and steadily the single stranded DNA will get converted further into double stranded DNA molecule. Okay,
This conformation of the template leaves only a single template base in the active site, preventing any confusion as to which base is ready to pair with the next nucleotide to be added.
okay, so which nucleotide is going to be added, so there would be no confusion among them. Okay, so that is why this is moving in a particular direction.
In contrast to the fingers and the palm, the thumb domain is not intimately involved in catalysis. Instead, the thumb interacts with the DNA that has been most recently synthesized. This serves to maintain the correct position of the primer template junction in the active site and reduce the rate of dissociation of the polymerase from the DNA substrate.
Now, the third domain, the palm domain. The palm domain is the third domain of the DNA polymerase 1 or the Conberg polymerase. This palm domain helps to hold the primer template junction in the active site. It will be holding the primer and the template junction. This is the primer, this is the template in a correct orientation. reduces the rate of dissociation of the DNA from the DNA polymerase. Now, this is it. DNA, it has been holded with the help of thumb domain and then will be made to close with the help of finger domain, forming a fist. और फिर वो मुक्का बन जाएगा, ठीक है? कैसे? क्योंकि डीएनए को होल्ड कर रखा है यहाँ पर अपने थंब से और यहाँ पर आप फिंगर से होल्ड कर देंगे, यू विल फॉर्म अ फिस्ट. ओके.
Another function of the palm domain is proofreading. The palm region makes extensive hydrogen bond contacts with the base pairs in the minor groove of the newly synthesized DNA. These contacts are not base specific, but they only form if the recently added nucleotides are correctly base paired.
Okay. So you can look here. There is a groove that is going to be produced, and the groove that is going to be produced here is referred to as the minor groove of the DNA molecule. This minor groove is being produced by the region of the palm domain. The palm domain is going to interact with the minor groove of the DNA molecule with several number of nucleotides. Okay, palm domain hydrogen bonds to base pair in the minor groove. Hydrogen bonds only form if nucleotides are correctly base paired.
In the rare event that a mismatched base pair is added, the distorted geometry of the substrate causes the replication rate to slow dramatically. The palm domain is now not able to make contacts with the minor groove, and consequently, the primer template junction is free to move about and interact with the exonuclease site.
Okay, so you can see this. There is another site present on the palm domain, and that site is referred to as exonuclease site. Okay, exonuclease site. Now, what is this exonuclease site? Exonuclease site is the one where the exonucleases are going to work. And once if a wrong nucleotide has been incorporated, then the DNA will move from the replicating site to the exonuclease site, because here now the movement of the DNA polymerase, that is Kornberg DNA polymerase, will be slowed down. When the movement of the Kornberg DNA polymerase will be slowed down, then what will happen? The DNA will be expelled from the DNA replication site. It will move to the exonuclease site where the wrong base will be hydrolyzed. Look here.
The exonuclease site removes incorrectly base paired nucleotides, starting from a 3' DNA end, and is therefore called a proofreading exonuclease. Removal of the incorrectly paired base is a matter of kinetics. With a mismatched base, the diminished replication rate is now slower than the exonuclease rate, and therefore excision of the mismatched base is greatly favored over polymerization on top of the mismatch.
If there has been a mismatch, then there will be a slow rate of movement of the DNA polymerase 1 or the Kornberg polymerase. In such a case, the Kornberg polymerase, from the Kornberg polymerase, the DNA will be expelled to a site that shows the exonuclease activity. Here, the nucleotide will be expelled out. The nucleotide has been removed, and now this DNA could again participate in the process of DNA replication.
After this potential mutation is corrected, the primer template junction slides back into the DNA polymerase site, and replication continues.
After this, once this replication has been completed, it then moves. Once this error has been removed, it then again moves to the DNA replication site where the process of replication again continues.
How well have you learned the mechanism of DNA synthesis?
This is all about DNA polymerase 1 and its activity in the case of the DNA molecule. Okay, if you have any doubt, now you can ask me. So now after finishing the DNA polymerase first, we will have to look at the DNA polymerase third. The DNA polymerase third represent the main replicating enzyme in the case of E. coli. Similarly, as the DNA polymerase third is the holo enzyme which is present in the case of E. coli, this DNA polymerase third is made up of many number of subunits. The polymerase third holoenzyme is a huge multi-enzyme complex that consists of 10 distinct polypeptides, and those 10 distinct polypeptides are alpha, omega, alpha, epsilon naught, theta, gamma, tau, delta, xi, psi, beta, delta, etc. The hallmark of the DNA polymerase third are its very high catalytic potency and processivity. So the hallmark are the DNA, are the catalytic potency as well as processivity. So this is the hallmark. Processivity refers to the ability of an enzyme to catalyze many consecutive reactions without releasing its substrate molecule. So the processivity that we know refers to the process of addition of large number of substrate molecule without releasing its substrate. Okay, processivity of an enzyme, when you talk about processivity, how many reactions an enzyme can catalyze without removing itself from the substrate molecule is referred to as its processivity. So we are going to look at the processivity. It is the ability of an enzyme to catalyze many consecutive reactions without releasing its substrate molecule. The enzyme extends the DNA chain with a very high processivity, and the highest processivity has been obtained from the use of this particular enzyme molecule. This replication complex is an asymmetrical dimer. Okay, there is a complex which is being produced of the DNA polymerase third, which is an asymmetrical dimer. And we are now going to look at the structure of the DNA polymerase third, how this DNA polymerase third is being produced, and what is its catalytic core, how does it controls the process of DNA replication, which are the different subunits, and how it controls the process of DNA replication as well as catalyze the polymerization activity. In what manner the polymerization activity is going to take place, and how and in which manner 3 prime to 5 prime and 5 prime to 3 prime, how are the different polymerase activities going to be processed. So for that we must look here. We must also look here at the dimer of the beta subunit forming a ring shaped structure and act like a sliding clamp. Okay, this is the structure of the DNA polymerase third. Okay, here you can find these are the different subunits that are being involved in the enzyme. So the different subunits which are being involved in the enzyme are as follows. Just look at this reaction, this complex. This is the complex that will have to study. Just focus here in the diagram. There is the leading strand synthesis going to take place with the sliding beta camp that is going to be synthesized continuously, and the other strand that is going to be synthesized discontinuously is through the lagging strand biosynthesis. Here the different subunits are the alpha, delta, epsilon naught, theta, tau subunits, and these subunits are going to interact together with each other. DNA polymerase third holoenzyme has been adapted from this diagram is from Levin. Now we are going to look at the functional counterpart of this enzyme complex. Okay. Now looking at its functional counterpart through animations. Okay. So let me look at DNA replication and move to the process of move towards the structure of DNA polymerase third. Okay, structural tutorial. Just a second.
The self-complementary nature of
Looking to share the same with you all. Okay, this is the animation of DNA replication. But what we have to see here is that we will have to see that
DNA
in what manner the process of DNA replication takes place with the help of the
in
DNA
bacteria.
polymerase third. So now we are going to look about the DNA polymerase third. In the case of bacterial cell, the two DNA polymerase, one working on the leading strand, another working on the lagging strand, are not the separate entities. They are same. They are being joined with regard to each other, and they are joined with the help of tau subunit. How?
The two DNA polymerases responsible for replicating the leading and lagging strands are linked together in multi-protein complexes called holoenzymes. How do two DNA polymerases remain linked and yet synthesize DNA on both the leading and lagging strand?
Now, how does the two nucleotides, how does the two DNA polymerases are going to be linked to each other? And in what manner they are going to be linked with regard to each other, that we will have to see. To look here, we must understand that a holoenzyme is a multi-protein complex in which a core enzyme activity is associated with additional component that enhances its function. This is the core enzyme activity, and then there is the dimerization component which is present, tau component. Okay, the other minor component are a part of this particular DNA polymerase only, and the sliding beta clamp will be loaded again and again over the tau subunit.
The trombone model explains how the replication machinery might exploit the flexibility of DNA to accomplish this task. According to this,
Now, there is a model which is being given in order to explain that how the DNA replication is going to take place. The model is commonly referred to as the trombone model of DNA replication. Now, what happens in the case of trombone model? I've already told you that trombone refers to as the rise and fall of a apparatus which was being used in order to provide relevant information to the sailors sailing. Okay, supposedly there is an ocean. Okay, there are ships which are sailing in the ocean, and there are sometimes big rocks or even mountains just beneath the surface of the ocean. Okay, just beneath the surface of the ocean there are mountains. Then, in order to protect our ship which is sailing across the ocean, there are techniques which have been developed. Which are the techniques which have been developed? Those techniques involve the tying of a bell at the towards with the with the tying of a ringing bell in the form of a trombone, along with the mountain. When the waves are going to move above or below, the trombone is going to move in both the directions. As the trombone moves, the bell is going to ring. As the trombone moves, as the trombone moves, as the trombone moves, the bell rings, the bell rings, the bell rings, and produces alarming sound, and produces alarming sound, alarming sounds. Then it is going to produce alarming sound, which will warn the sailors. So, this rise and fall of the trombone is exactly similar to the rise and fall that is going to be seen in the case of the lagging strand of the DNA molecule. As the rise and fall in the case of lagging strand of the DNA molecule is going to be equivalent to the rise and fall of the trombone. So there you find that such type of model will be referred to as a trombone model. Okay, look here.
this model, the single-stranded DNA template for the lagging strand pulls through the DNA polymerase, similar to the slide of a trombone. This flexibility allows the DNA polymerase to add nucleotides to the 3' end of the growing lagging strand.
Okay, now once the nucleotides are primer is going to be added, then see this, As
The
the
DNA
DNA,
polymerase
just a
haloenzyme
second, as the DNA is being biosynthesized continuously in the leading strand, at the lagging strand, because of the opposite orientation of the other DNA polymerase, the lagging strand is again and again being dissociated and again joined over the DNA molecule. लैगिंग स्ट्रैंड को बार बार डिसोसिएट किया जा रहा है और दोबारा से ज्वाइन किया जा रहा है ताकि लगातार डीएनए की बायोसिंथेसिस की जा सके. Because the lagging strand is being biosynthesized in opposite direction, so it is being dissociated again and then again and again reassociated in order that the DNA biosynthesis can take place in a continuous manner. Such type of model is commonly referred to as trombone model of DNA replication. Okay, so with continuation to that, we are moving forward.
also contains a sliding clamp loader. This
Now
multi
this is the sliding clamp loader. This sliding clamp loader is the one which will be loading the DNA molecule with the primer attached over itself again and again over the lagging strand so that the new strand could be biosynthesized.
multiprotein complex loads sliding clamps onto the primed DNA. The sliding clamps help the DNA polymerase find the primed DNA and then help increase the processivity of the DNA polymerase.
Okay, it is this sliding clamp that is going to increase the processivity of the DNA polymerase, that is going to bind to the DNA molecule and then increase the processivity continuously.
Do you understand the mechanism of replication well enough to keep a cell alive?
Now, see this. This is what we have to understand about the process. Now, moving forward again to the pathfinder that we were studying. Okay, so now this diagram clear to you all, as well as the components, its functioning, as well as in what manner it is going to be functional. Now, please look here. We are going to talk about the DNA polymerases, how DNA polymerases are going to function. The enzyme DNA polymerase can be categorized into three different types: DNA polymerase 1, DNA polymerase 2, and DNA polymerase 3. These are the three different types of DNA polymerases that are being associated. DNA polymerase 3 is just made up of 10 different subunits. It is the main replicating enzyme which is being involved in the process of DNA replication. DNA polymerase 1 is involved in DNA repair, gap filling. DNA polymerase second is involved in DNA repair. Okay. Now, the same type of things are going to take place in the case of eukaryotes. You have already seen this structure. Now, please look here. The 5' to 3' exonuclease activity of polymerase 1 is independent to 3' to 5' exonuclease activity. Now, see, there is only one DNA polymerase in the world which has both the exonuclease activity 5' to 3' as well as 3' to 5' exonuclease activity, and that is referred to as DNA polymerase 1. Okay, if you are going to remove a single exonuclease activity from the DNA polymerase 1, then it will be referred to as the Klinov fragment. Okay, the 5' to 3' exonuclease activity of the DNA polymerase 1 is independent to 3' to 5' exonuclease and its polymerase activity. Proteases such as subtilin and trypsin cleaves polymerase 1 into two fragments. So if you are going to cleave the exonuclease with the help of proteases, then it is going to cleave into two different fragments. One is called as 3' to 5' exonuclease fragment, and the other one is 5' to 3' exonuclease activity. So these are the two different fragments which are going to be produced. Okay, any doubt? Any problem? Now, the one fragment that would be removed, then the other fragment that would be produced is referred to as the Klenow fragment. Okay, there are two fragments which are being produced: a large C terminal or the Klenow fragment and the N terminal fragment. This fragment which is going to be produced has the 3' to 5' exonuclease activity, which is similar to all the polymerases. See, each and every polymerase has 3' to 5' exonuclease activity. You can look here. Each and every polymerase has 3' to 5' exonuclease activity. See this? Everyone has this type of an activity present within itself. You can look here. So it is present in all. Whereas this activity 5 prime to 3 prime is only present in the case of DNA polymerase third. DNA polymerase 1, only DNA polymerase 1 has 5 prime to 3 prime exonuclease activity that is present. Okay. So this is going to be removed with the help of treatment with pepsin or trypsin. Then it would be used in Nick translation. Now, which type of question is being asked in Nick translation? Please look here. See, if there is a DNA molecule, okay, there is a gap that is going to be produced. There is a nick that has been produced in this DNA molecule. First, you are going to repair this nick with the help of DNA polymerase 1, DNA pol 1. Then, in the same case, in the same DNA molecule, when you are taking the same DNA molecule, okay, you take the same DNA molecule, okay, the same DNA molecule has been taken. And when the same DNA molecule has been taken, now you are going to cleave the same DNA molecule with the DNA polymerase 1, that is referred to, we are going to repair it with DNA polymerase 1, that is referred to as Klenov fragment. Klenov fragment. Then what is going to happen? Then the repair is going to take place. What would be the difference that would be produced? Please note it down that in this particular case, not only the DNA will be repaired, but the DNA segment that is going to be repaired is also going to be replaced because it has an exonuclease activity now. So it will replace all the existing nucleotides. All the nucleotides will be broken down. All such nucleotides will be broken down. They would be cleaved, and new nucleotides being biosynthesized. So this is the dsDNA. Okay, this is the dsDNA produced. Now there is a cleavage which has been produced. Now please look here. If there is a Klinov fragment, then nothing is going to happen. Only some nucleotides are going to be added and nothing more is going to happen. So what will happen? Only few nucleotides will be incorporated, just like this. This is the major difference that is going to be produced. The first question in the case of T.A. Brown book of RDT is about this Kornberg polymerase, and this question has been asked in CSIR net JRF entrance examination, that how does DNA polymerase 1 and Kleno fragment, the fragmented DNA polymerase from which the 5 prime to 3 prime exonuclease activity has been cleaved, has been removed by treatment via treatment with proteases such as pepsin or trypsin. Okay, then this is going to be produced. Any query, any question, you just ask me. Any query, any question, you just ask me. No query, I can move forward. Okay. So then now we will have to start about the eukaryotic DNA polymerase. How does eukaryotic DNA polymerase are going to function, and what about the eukaryotic DNA polymerase? So we know that in the case of eukaryotes, the DNA polymerase will be divided into several different types. That would be alpha, lambda, delta, epsilon naught, and kappa. DNA polymerase alpha, lambda, delta, epsilon naught, and kappa. Okay, these are the DNA polymerases that we are going to function in the case of eukaryotes. So from tomorrow, we are going to start from here. Okay, this is all for today. Hope you enjoyed the lecture. We will continue and finish this process that has been done. The entire mechanism has been taught to you. This entire mechanism has been illustrated from in the animation that has been provided to you, that how the incoming nucleotides are being attacked by the oxygen atom, and it will attack the innermost phosphate molecule in a nucleophilic acyl substitution reactions. We have finished elongation, would be finishing initiation, elongation, and termination. This is the trombone model, which I have already explained. Tomorrow, we are going to finish this particular process of DNA replication. So please just at least give it a reading and try to solve the questions in the MCQ part, as well as look at the animated video. I would be sharing the same with you in the group. That's all for today. Hope you enjoyed the lecture. That's all. Good night. Bye bye.